Hello, I'm Alejandro Cifuentes. I lead the Foodomics laboratory at the National Research Council in Madrid. It is a real pleasure to present the topic of Foodomics and discuss its application in food science and nutrition. Omics sciences share the global aim of identifying, describing, and quantifying the biomolecules and molecular processes that contribute to the form and function of cells and tissues. As applied to food, Foodomics is a discipline that studies the food and nutrition domains through the application and integration of advanced omics technologies to improve consumers' well-being, health, and knowledge. To start explaining the concepts of omics and foodomics, it is essential to understand the central dogma of biology that describes the flow of genetic information within biological systems. We know that DNA is the type of molecule in which all the biological information is stored. DNA is transcribed to RNA, and this RNA is translated into proteins, which are essential for cell function and structure. The biomolecular products resulting from the activities of these proteins are known as metabolites. This central dogma of biology can be applied as a framework to link different biomolecules to the associated omics technologies that are employed in Foodomics. For instance, genomics is used to investigate the genes formed by DNA. We are using transcriptomics to investigate RNA. We are using proteomics to investigate proteins, and we are using metabolomics to investigate the metabolites. Moreover, glycomics is used to study carbohydrates, lipidomics is used to study lipids, and ionomics is used to study minerals and elemental ions. Foodomics uses advanced analytical technologies in a high-throughput manner to study these diverse food components. In food science and nutrition, we are moving from very reductionist studies from which we were using targeted analysis to determine whether a given compound was present or not in a food, to holistic approaches that allow us to discover biomarkers and other biomolecules towards addressing more complex issues related to food science, food composition, nutrition, and the effect of foods on our health. Only 150 or fewer nutritional components are tracked in food composition tables. While the food components that have historically been measured are essential for nutrition, this food composition data provides a limited representation of the vast diversity of biomolecules that are in food. As society transitions from inadequate amounts of food to inadequate quality of food associated with diet-related diseases, the integration of Foodomics approaches to characterize food composition can be tremendously transformative. We are seeing a tremendous expansion in our knowledge of the components in food with Foodomics that complement traditional approaches in nutrition and food sciences. For example, a search done in the periodic table of food initiative, the PTFI database, which provides data using standardized Foodomics tools, showed the vast diversity of components in food. This knowledge tremendously expands our understanding from the back of a nutritional level. Diverse data users can variably apply this data for designing food system solutions, from breeding crops with higher nutrient density to identifying plant-based foods with similar composition as animal-based foods. This visual shows the integrated approach necessary to carry out a Foodomics study as part of a nutrition intervention. You can see that the vast diversity of biomolecules and other components we aim to identify requires a broad range of tools. This includes sequencing and different separation techniques, all of them coupled to high-resolution mass spectrometry, the most common tool in omics studies. In high-resolution mass spectrometry, all samples, whether liquid, solid, or gas, have to be passed to the gas phase and ionized. Then, the ion is analyzed in this high-resolution mass analyzer and finally detected. These are the basic parts of any mass spectrometry instrument, regardless of what type of Foodomics platform we are using, such as metabolomics, proteomics, lipidomics, ionomics, or other omics. Using this high-resolution analysis, we can identify different types of components in foods based on their different molecular mass, isotopic profiles, and fragmentation patterns, going from small metabolites to proteins. Even more, we can identify many of these compounds in a single analysis, which was not possible following a more traditional targeted food analysis that characterized much of food and nutrition science. Data integration, bioinformatics, and artificial intelligence combined with Foodomics data are enabling new approaches to make sense of the incredible amount of data that is now generated and the biological information this data can provide. There are many different possibilities for how to combine technologies, equipment, and approaches to measure the foodome, although we still have to overcome many bottlenecks, including the need for more standardization across the field. Foodomics is well positioned to help address many of the pressing challenges of the 21st century, including food security, food and biodiversity loss, food safety, new contaminants and new pathogens, food waste and food loss, food bioactivity, climate change, and diet-related chronic disease. Addressing many of these societal challenges will require a more comprehensive and holistic understanding of biological systems, including dynamic interactions instead of the traditional reductionist approach that has dominated food sciences and nutrition. Let's take an example regarding food bioactivity and health. Right now, there is a shift in research from the prevention of deficiency diseases toward the prevention of chronic diseases, with elucidation of the role of food constituents on such diseases. The relationship between food intake and chronic diseases is much more complex, necessitating a more holistic approach to understanding how both the essential and non-essential food constituents interact with other systems in the body to prevent the disease. As an example of the Foodomics work we are carrying out, Alzheimer's disease is a multifactorial pathophysiology that originates from neurodegenerative impairment. Alzheimer's disease is responsible for 60–80% of the dementia cases in older people. A critical problem of Alzheimer's is that no cure has been found for this illness yet. So, in our laboratory, we are investigating if diet can prevent the apparition or reduce the development of Alzheimer's disease. And I can tell you, we have obtained interesting results with different bioactive components from different natural sources. We have demonstrated this promising neuroprotective potential using in vitro and in vivo experiments with animals, but still we have to corroborate it using clinical trials with humans. This work brings to light the crucial role that Foodomics can play in our understanding of the complex nexus of food science and nutrition domains, including the binomial food and health. In summary, Foodomics is enabling the required shift in food science and nutrition research from a reductionist approach to a systems approach to better characterize food composition and to enable leveraging resulting data to address the interconnected challenges of our times. The 21st century is observing a technological revolution that includes breakthroughs in nutrition, genetics, omics, including Foodomics, informatics, including bioinformatics, global networking, remote sensing, precision nutrition, precision farming, low-impact agriculture, sustainability, biorefinery, and One Health, among others. These advances will hopefully continue to drive major global solutions for society and in our daily life. To finish, what I can tell you is that Foodomics will help us to improve our knowledge about food science and nutrition. But what I can also tell you is that we have a long way to go. But no doubt, the road ahead will be exciting and the reward worthwhile. So let's keep walking together.